Hi class, welcome to our lecture on metabolism. Metabolism is a very fascinating subject. It's also quite a complex subject. So I'm going to be doing my best to explain it to you in a way that is easy to understand. All right, so what is metabolism? Metabolism refers to the chemical processes involved in maintaining life. And metabolism allows us to release energy from foods that we eat, carbs, fat, proteins, and alcohol, and allows us to synthesize new substances when we need them, as well as get rid of substances when we don't need them. The metabolic pathway is a group of reactions that occur in a progression, and many intermediate products are formed during metabolism. So there are two kind of routes through metabolism. There are anabolic pathways and catabolic pathways. Anabolic or anabolism means building reactions, and catabolism is breaking reactions. So in anabolic pathways, we are trying to build bigger compounds. Remember, we can use glucose as the building block for polysaccharides. We can use fatty acids to make triglycerides, cholesterol to make some of our hormones, and amino acids to make large proteins. This process uses energy, and it makes sense that we would be doing more of this when we are growing. Catabolic pathways are breaking reactions. So the example here is glycogen being broken down to make glucose. This could happen when you have low blood sugar levels and you need that glucose to get to your cells. We could also break down large proteins into single amino acids so that those amino acids could be reassembled into something that we needed. In catabolism, it results in the release of carbon dioxide, water, and energy in the form of ATP. And this pathway is more predominant during weight loss or wasting. So how do we convert food into energy? Well, we do so through metabolism. And converting food into energy, remember energy is ATP for us, is a catabolic reaction. So this is a breaking down reaction, breaking down that hamburger or that corn on the cob or that piece of pizza into something our body can actually use. So we send monosaccharides, amino acids, fatty acids, and glycerol, as well as alcohol through our metabolic pathways, and we can capture energy as ATP. ATP is the body's form of energy. It's kind of like our currency for energy. Heat, carbon dioxide, and water are also released in this process, as we said on the last slide. These are a couple videos you can watch that help to explain some of these reactions, and I'll put the links on the web as well. All right, so in a nutshell, an overview of catabolism. There are three main stages. The first stage is digestion, breaking the complex molecules to their building blocks. Then conversion of building blocks to acetyl-CoA, which we're going to talk about. This is a very important molecule. And thirdly, metabolism of acetyl-CoA to carbon dioxide and formation of ATP. So depending on what type of food we eat, whether it's a carb, a protein, or a fat, or a mixed food, energy will be created through slightly different pathways. And I mentioned that acetyl-CoA is very important. Acetyl-CoA is this compound right here in the center of this all. And acetyl-CoA is something that can enter what we call the citric acid cycle and then can produce electrons to go into the electron transport chain and can generate energy. So all different types of foods can be converted to acetyl-CoA. What is this energy we've been talking about? It is ATP. So ATP is the main energy source for the body, and only ATP can be used to make new compounds, help muscles contract, conduct nerve impulses, and pump ions across membranes. It's very, very vital to life. ATP is made out of an adenosine bound to 3 phosphate groups. So here you can see the adenosine right here, and then 3 phosphate groups. And there's arrows indicating high energy bonds. So these phosphates can be separated 1 phosphate at a time, and each time you break off a phosphate group, energy is released. This is another picture of ATP and how that phosphate group can be broken off. So here we have ATP. It's got 3 phosphate groups. When we break it down, it forms ADP plus P, this free phosphate group. And we can use some of that for energy. ATP is high energy and ADP is low energy. If we want to convert ADP back to ATP, we can. We need to add that phosphate group back. However, it requires energy to do this. Cells are constantly breaking down ATP and regenerating it, and ATP synthesis involves the exchange of ions in the form of hydrogen ions from energy-yielding compounds. All right. So, ATP produced. So cellular respiration happens when food molecules are broken down to form ATP, and oxygen is needed as the final electron acceptor. This can be an aerobic or anaerobic process, and aerobic means in the presence of oxygen. It is much more efficient and can generate much more energy, many more molecules of ATP. Anaerobic is without oxygen, and this can generate some energy, but it's not nearly efficient as aerobic respiration or aerobic cellular respiration. So first we're going to talk about aerobic respiration. There are 4 stages of aerobic respiration. There is glycolysis, a transition reaction, the citric acid cycle, or the electron transport chain. And we're going to talk about each of these in detail on the upcoming slides. So the very first step of aerobic respiration is glycolysis. And if you think about this word, the very first part of this word is glyco, and then the last part of this word is lysis. And the first part indicates you're going to be breaking down glucose and lysing it, breaking it. Lysis is breaking it. So glucose, converting glucose into 2 molecules of pyruvate. And pyruvate is a very important 3-carbon compound that is used in metabolism. Glycolysis is the first step of aerobic metabolism, and it takes place in the cytoplasm of the cell. Afterwards, aerobic metabolism moves into the mitochondria of the cell. All right, so what is important for this all to work? There are quite a few important ingredients, you could say, or necessary factors for metabolism in metabolic cycles. And some of them happen to be vitamins that we need that are essential to our diet. So niacin and riboflavin actually are used to transfer hydrogen ions from energy-yielding compounds to oxygen in metabolic pathways. Remember that oxygen is the final electron acceptor. Niacin functions as a coenzyme, which is something that is needed for an enzyme to function. And so niacin in metabolism is in the form of nicotiniamide adenine dinucleotide, or NAD, and then it can accept a hydrogen ion to become NADH, nicotiniamide adenine dinucleotide hydrogen. Riboflavin plays a similar role, and in its oxidized form, meaning without hydrogen. It is FAD, flavin adenine dinucleotide. In its reduced forms, it actually accepts 2 hydrogen atoms. So reducing oxygen to form water is the driving force for life. And what reduction means is adding these hydrogen atoms to oxygen, because remember, water is H2O. So that is part of how it is done. So NAD and FAD, niacin and riboflavin are important, very important compounds that help us make energy. Wow, what is going on here? All right, so over here, this is just another picture summarizing aerobic metabolism. So over here is glycolysis. And remember, glycolysis starts with glucose and it ends with pyruvate. And it ends with 2 molecules of pyruvate, so we got 2 times here. It also produces water and it produces ATP. We've got an ATP there, an ATP there. We've got 2 NADHs here, but we're using some ATP here. So we are using ATP, but we're also producing ATP. So you can see at the bottom half of this, you have 2 ATP plus 2 ATP. So you have 4 ATP gained in this last half of glycolysis, where the first half uses 2 ATP. So the net ATP that is made during glycolysis is 2. Remember, this happens in the cytosol, beautiful writing here, of the cell. And then the reaction moves into the mitochondria, where it enters the citric acid cycle, otherwise known as the Krebs cycle, and then moves along to the electron transport chain. So this was just another picture to explain the concept that we're going to see in detail. All right, glycolysis. So we just saw what glycolysis is. Glucose is oxidized to form 2 molecules of pyruvate. Pyruvate is a very important compound that has 3 carbons. NADH plus H, or NADH2, is produced, and 2 net molecules of ATP, because we had to use 2 in the beginning and we produced 4 in the last half of glycolysis. This happens in the cytosol, and this is how we break down our carbohydrates to produce energy. This is great because it does not require oxygen. So there's no oxygen needed yet as the final electron acceptor, not yet. Wow, okay, so this is glycolysis explained in much greater detail. And here you can see these blue circles represent carbons, yellow is phosphates, and then you have ADP and ATPs here. So when you guys take chemistry or metabolism class, you will be responsible for memorizing all of the names of these different intermediates in this pathway. However, for this class, mostly what I want you to know is that glucose is converted to 2 molecules of pyruvate. Pyruvate is right here. I'm not going to write it because my writing is so bad. Or I could do this, glucose to pyruvate. And you have some byproducts that are helpful. You've got NADH plus H, and you've got some ATP happening. So that is the main thing to know about glycolysis. Transition reaction. This is the second step in aerobic respiration. So the transition reaction is a reaction that converts pyruvate, a 3-carbon compound, to acetyl-CoA, which is a 2-carbon compound. And this is necessary because acetyl-CoA can enter the next step in metabolism, the citric acid cycle. This is irreversible, so once this happens, you cannot go back to glucose, and this requires oxygen. So glycolysis did not require oxygen, but this step does. This is a picture of what happens in this intermediate step, the conversion of pyruvate to acetyl-CoA. A carbon is lost, CO2 is created, NADH plus H is created. All right, citric acid cycle. So we have to convert pyruvate to acetyl-CoA in order to enter the citric acid cycle. And remember, the citric acid cycle is the third step in aerobic respiration, and it takes place in the mitochondria. There are other names for the citric acid cycle. I have seen tricarboxylic acid cycle and Krebs cycle as well, but our book refers to it as the citric acid cycle. Once a molecule of acetyl-CoA enters the citric acid cycle, it combines with something called oxaloacetate and goes around the citric acid cycle. It goes around one time for every acetyl-CoA, but remember that we made 2 pyruvates from glucose, and so therefore we can have 2 acetyl-CoA, and therefore we can go around this cycle 2 times. So what is produced by this cycle? Energy in the form of NADH, FADH2, potential ATP, and carbon dioxide, and this does not require oxygen. All right, so here it is. This is the citric acid cycle in pretty good detail. So up top, we've got acetyl-CoA. Acetyl-CoA combines with oxaloacetate, which is pretty important for some of my later discussions in this chapter. But it's a 2-carbon compound that combines with a 4-carbon compound. And the very first step in the citric acid cycle is you have a 6-carbon compound. So then this 6-carbon compound moves through the cycle. And again, for this class, you do not have to memorize itsy bitsy details, but I want you to know the main things. How energy is produced, what type of energy is produced, and how many turns of this pathway can occur based on one glucose molecule. In your appendix, there are even more detailed steps about this. Okay, so oxidative phosphorylation. So this is where we take energy from NADH and hydrogen and FADH2 and use that to form ATP. We can do this by transferring those hydrogen molecules or oxidizing these compounds. So NADH plus H is oxidized to NAD plus, and FADH2 is oxidized to FAD. So you can see both of these are giving away 2 hydrogens. They're giving away 2 hydrogens, and those hydrogens combine with oxygen to form water. So here is where oxygen is key for metabolism. This happens in the electron transport chain, and this is what it means when we say oxygen is the final electron acceptor, because if oxygen wasn't there, it couldn't accept those hydrogens, and we couldn't make that water and make that ATP. So in the electron transport chain, you can generate 2.5, I'm going to write this, so you can generate 2.5, this is the point, 2.5 ATP for each NADH plus H and 1.5 ATP from an FADH2. We also had that molecule of GTP, which you saw back here on this diagram, and that GTP can be used to turn an ADP into an ATP. So we're making energy out of these non-ATP compounds. We're making ATP from them. The electron transport chain can make 28 ATP, and so this is where most of the energy is created. And this is the fourth step in metabolism. All right, so this is it kind of expanded in a nutshell. So here we have got glycolysis happening up top right here, glucose to pyruvate. This is our first step. Then we've got pyruvate to acetyl-CoA, our transition reaction, that's our second step. We've got the citric acid cycle, our third step, and then we've got our electron transport chain, our fourth step. And I don't actually love this diagram, but it is showing that NADH and FADH2 can be used to create ATP. Wow, what is this? So this is a diagram I got offline, and again, this is more complex than you need to know, but I do like it because what it's showing here is NADH plus H and FADH2, and they are moving through these electron carriers, or they're going through reactions where they're losing their electrons, their hydrogens, and those hydrogens are being pushed through these electron carriers in the cell. So as the electron transport chain continues, you have this buildup of hydrogen ions out here. And that's really good because we can use those for energy. However, we can only use them for energy if water is, or if oxygen is available. Here you have something called the ATP synthase, and the ATP synthase can use the energy from these hydrogens to convert ADP back to ATP and make energy. All right, so oxygen, I've been hinting that it's important. I've been saying that it's important. So without it, those hydrogens would build up, metabolism would stop, and we would actually die. It is absolutely crucial for life, and this is a good video explaining the difference between what happens when we have oxygen versus when we don't. And I will play this, put a link to it on Polylearn. All right, so we talked about aerobic metabolism occurring in those 4 steps. Now we can talk about anaerobic metabolism. And anaerobic metabolism is not as effective as aerobic metabolism because it converts only about 5% of the energy in a molecule of glucose to energy that's stored in high energy phosphate bonds. Remember, ATP has those high energy phosphate bonds. When would this happen? So this might happen if a cell doesn't have a mitochondria, and there are some bacteria cells that don't have mitochondria. Remember, those last parts of metabolism took place inside the mitochondria. So if you don't have a mitochondria, how are you going to run the electron transport chain? This could also happen if oxygen is lacking. For example, if you are doing strenuous exercise and really short of breath, or in a very bad scenario, if you were suffocating. But let's just say you're running a marathon and you're trying to break a record. You're running as fast as you can and you're pretty short of breath. So what happens here? So we have glycolysis as our first step in anaerobic metabolism, because remember, glycolysis did not require oxygen, so we can still make pyruvate. However, that transition reaction from pyruvate to acetyl-CoA did require oxygen. So in the absence of oxygen, pyruvate is converted to lactate. And it's converted to lactate by an enzyme called pyruvate dehydrogenase. And this step actually uses some of the energy. Lactate will be produced by red blood cells. It can be picked up by the liver and used to synthesize pyruvate. All right, so this is the diagram of how glucose can be used to make lactate in the absence of oxygen. So in this top half, we have glycolysis, as we've seen it before, and then this bottom half, we've got pyruvate to lactate. But you can see pyruvate to lactate is using energy instead of making energy. Once pyruvate is converted to lactate, lactate actually goes to the liver, to something what's called the Cori cycle. So lactate is going to the liver. So here's our liver. And in the absence of oxygen, our muscle cells will begin to produce lactate, because remember, we've got glycogen stores in our muscles, can break down that glycogen to glucose. Glucose we can convert to pyruvate via glycolysis, but then pyruvate we cannot convert to acetyl-CoA. So instead we convert it to lactate. So this will happen in our muscle cells. Lactate can leave the muscle via the bloodstream, and it can enter the liver. And then liver enzymes can actually convert lactate back to glucose, but this uses energy. Once the liver has converted lactate back to glucose, glucose can be given back to the muscle cells. So you can see this is kind of an inefficient pathway. I would say it's a very inefficient pathway. So even though our muscle cells do have mitochondria, when we're working out really intensively and we're not able to catch our breath, they have to rely on anaerobic metabolism. And so they begin to create lactate. All right, so that is kind of carb metabolism in a nutshell. And we're going to talk about fat and protein metabolism. I guess I want to say one more thing about lactate, though. Once the lactic acid builds up in the muscle, it can cause muscle fatigue and oxygen debt, and eventually the muscles will cramp and will stop working. So you may have even felt some symptoms of this during a high-intensity workout. Once you bring that intensity down, get oxygen to those muscles again, this can usually clear up. So what would you think fat breakdown produces? Would you think it makes more or less energy than carb breakdown, remembering what you know about fats? I'm hoping that you thought really hard and used your critical thinking skills like smart students and thought, okay, well, fats have 9 calories per gram, carbs have 4, so I think fats make more energy because energy is our ATP. And if that's what you thought, you guys are so smart and I'm so proud of you. You're right. So how do we start this? We start this with something called lipolysis. And anytime you see the word lysis, it means breaking down. And what comes before that is usually what you're breaking down. So we're breaking down lipids. We can break down triglycerides, which remember are our main source of fats and our circulating source of fats in our blood. And we break those down into glycerol, which is the backbone, and fatty acids. This all happens in the mitochondria. So next we can break down those fatty acids for energy production, and fatty acids will donate electrons to oxygen. The fatty acids are brought to the mitochondria by a molecule called carnitine, and there's actually a carnitine shuttle that helps the fatty acids get inside the mitochondria. So here is what I just said in a diagram. In our adipose tissue is where we have our triglycerides stored. We also have some circulating triglycerides in the bloodstream. We can break down these triglycerides into monoglycerides, glycerol and then also glycerol and just single fatty acids. This is done by a hormone called hormone-sensitive lipase. So this lipase hormone breaks down the triglycerides to the single fatty acids. Fatty acids are taken up by the cells and they are shuttled to the mitochondria by carnitine. So remember, fatty acids are made up of even number of carbons and they transfer their energy to carbon groups at a time. So so 2 carbons at a time. So acetyl-CoA When we cut off 2 carbons from the fatty acids, we can make acetyl-CoA. And hopefully you remember that acetyl-CoA goes directly to the citric acid cycle, and it will combine with oxaloacetate. We can also create NADH plus H and FADH2, and those we can use to convert ADP to ATP and yield energy. This is referred to as fatty oxidation or beta oxidation. I want to add a word in here about long fatty acids versus medium chain fatty acids. You may have heard of medium chain fatty acids, and these are found in things like coconut oil. These are usually 6 to 8 carbons long, and because they are shorter, they provide less energy. They actually provide 8.3 calories per gram versus 9. Also, they don't require carnitine to transport them, and they don't have to be transported through the lymph. So those are pretty interesting. All right, so what is happening here? Wait, I want to go back and say one more thing. Okay, one more thing about fats. So this is straight from your book, but it's really important. So the big difference between glucose and fatty acids is that fats have lots of carbons. Remember, the average fat was like 18 carbons long. And then we have fats that are 20 carbons long, 16 carbons long, where glucose only has 6 carbons. So when you cleave off 2 carbons of fatty acids at a time, and each 2 carbons can make 1 acetyl-CoA, that's a lot of acetyl-CoA that you can create from fat, where glucose, you can only break it down to make 2 acetyl-CoA. So glucose can only go around the electron transport chain 2 times in its acetyl-CoA form. However, fatty acids might make many acetyl-CoA, like 5 acetyl-CoA or 6 acetyl-CoA, and that can help generate a lot more energy. Also, fatty acids have fewer oxygens than glucose, and fatty acids have more hydrogens. Glucose exists in a more oxidized state, meaning there's more oxygen on glucose. So that's part of the reason why fats make more energy. Okay, so this diagram is showing how fatty acids are broken down 2 carbons at a time, and how they can then enter the citric acid cycle and be used to create energy. So you start with a 16 carbon fatty acid, 2 carbons are cleaved off to make acetyl-CoA. Now it's a 14 carbon fatty acid, 2 carbons are cleaved off. Now it's a 12 carbon fatty acid. All right. So the problem that we run into with fat metabolism is we are making so much acetyl-CoA that sometimes we run out of oxaloacetate. So in the beginning, when I was talking about the citric acid cycle, I said, remember this oxaloacetate, this is going to be important. In order for acetyl-CoA to enter the citric acid cycle, it has to combine with oxaloacetate. So remember, we have the 2 carbon acetyl-CoA combining with the 4 carbon oxaloacetate, and they combine to form citrate, the first 6 carbon compound of that citric acid cycle, which then moves through the cycle generating energy. If you are short in oxaloacetate, acetyl-CoA cannot enter the citric acid cycle. So when we're breaking down fats, we end up sometimes using all of that oxaloacetate, and acetyl-CoA can build up. So what can happen is we need pyruvate, because we can convert pyruvate to oxaloacetate in a pathway that you guys haven't seen yet. But in order to get pyruvate, we need glucose or carbohydrates. So when we are eating a lot of fats, if we are not eating any carbs, eventually our metabolism can kind of slow down and we won't be able to break down fats as effectively. But if we add some carbohydrates, those carbohydrates can be used to regenerate oxaloacetate, which will ensure that the citric acid cycle is still able to run. So this is what I just mentioned in a diagram form. So here we have got fats. So we've got fatty acids entering the citric acid cycle, lots and lots of them as acetyl-CoA. But in order to do that, they need oxaloacetate. Oxaloacetate does not come from fats. It's got to come from pyruvate, which in this case is made from glucose. So in the presence of carbohydrates, the breakdown of fats actually works a lot better. When you have fats in high amounts, being able to break them down is more efficient with carbohydrates. Okay, so when these conditions are not met and you don't have that oxaloacetate, that acetyl-CoA has to go somewhere. So you're breaking down fats, you're breaking down fats, you're breaking down fats, you're building up acetyl-CoA. Where is that acetyl-CoA going to go? And this circumstance usually happens when people are eating a no-carb diet, a carb-free diet, or are fasting. And so what will happen is kind of this formation of what they call ketone bodies. So when there is no oxaloacetate to bind with acetyl-CoA and make citrate, then acetyl-CoA will actually be converted and combined to make ketone bodies. And there are 3 different types of ketone bodies. They're called acetoacetate, acetone, and beta-hydroxybutyrate. And 2 of these are very acidic, and they can cause the acid levels in our blood to build up, and something called ketosis will happen. So ketosis is when you have too many ketone bodies, which cause too much acid in your blood. So when would this happen? This would happen if there is insufficient insulin production. And when are you not going to make enough insulin? You're not going to be making insulin if you don't eat carbs, or you're not going to make insulin if you are a type 1 diabetic. So this can happen in type 1 diabetics. When it happens during fasting, it is a little bit less severe than when it happens in type 1 diabetics. In type 1 diabetics, this is very commonly responsible for hospitalization, coma, and even death. So you don't have insulin, and because of that, you start to break down your fats. That's great. That can work for a while. But you're going to be breaking down these fats, and eventually acetyl-CoA is going to build up. If it can't enter the citric acid cycle, it is recombined and converted to ketone bodies. And ketone bodies are 3 to 4 carbons long. And ketosis can occur. So other cells can convert ketones back to acetyl-CoA, which is good. This actually allows us to use our fat for quite a while. And our body will adjust to ketones over time to an extent. But like I said, this can be quite dangerous in diabetics. One of the ketone bodies, acetone, has a fruity scent, and you can actually expel it during your respiration. So you can do tests for ketone bodies in your breath. What do the ketone bodies look like? This is what the ketone bodies look like. And so acetyl-CoA is made because of this fat breakdown. But acetyl-CoA will combine to make these ketone bodies. And some of our muscles can use ketone bodies. So our heart, our skeletal muscles, and our kidney can use ketone bodies. But our brain really, really likes glucose. Okay, so in type 1 diabetes, I said this is really serious. If there's no insulin, the cells can't use glucose because there's no oxaloacetate available in that citric acid cycle to accept acetyl-CoA. Rapid fat breakdown will occur. Again, insulin will not allow glucose to get in the cells is a better way of saying that. You don't run into the oxaloacetate problem till a little bit later, till you're in lipolysis. So you start breaking down your fats instead. Acetyl-CoA will build up because of oxaloacetate, ketone bodies will be produced. I said they are acidic. We spill them into the urine. We try to get rid of them. However, unfortunately, to balance them out, it pulls sodium and potassium into the urine, which are important electrolytes. And we can have electrolyte imbalances. We can also become dehydrated. Our blood can be acidic, and it can even cause death. So the treatment for this is insulin, so that we can get glucose to the cells, and electrolytes if there's electrolyte imbalances, and fluids if there has been a lot of fluid loss. In semi-starvation. So in semi-starvation experiences, hopefully you don't do these on purpose, but you will have a similar reaction like a type 1 diabetic, but slightly less severe. So your glucose levels will fall, your insulin levels will fall. Heart, muscle, kidneys, and eventually the brain. I said the brain does not like ketone bodies, but eventually, as a survival mechanism, our brain adapts and is able to use ketone bodies. This spares proteins from being broken down. Our ability to use fats as energy for our vital organs, the heart, the muscle, the kidneys, and the brain, allows us to not have to break down proteins. We don't want to break down proteins because proteins are what keeps all these vital organs together, helps us make our blood cells, our immune factors. And when we lose too much protein, we actually die. So when you lose about 50% of your body weight in protein, you can actually die. Protein metabolism. So protein metabolism is happening in the liver. We just talked about fat metabolism. We were saying we don't want to break down our proteins, but what if we have to? If we break down our proteins, we break down our proteins in the liver. Some of them can be metabolized in the muscle, but these are only the branched chain amino acids, which are leucine, isoleucine, and valine. And so these branched chain amino acids are pretty popular supplements for bodybuilders. The very first step in breaking down a protein is called deamination, and you are removing that amine group or that nitrogen group from the amino acid. And so that nitrogen group then will go to the liver and the kidneys to be excreted in the urine. And we're going to talk exactly how that happens. The carbon skeleton that's left over after you've removed the nitrogen group can go to form ketone bodies, can go to oxidation, or can go to create glucose. So, I thought that I had something different on this slide, but I don't think that I did. Oh wait, I did. Okay. So gluconeogenesis. So this is the formation of glucose from non-glucose products. And gluconeogenesis happens when we convert protein to glucose or amino acids to glucose. There are only some amino acids that can be converted to glucose. Some amino acids are called glucogenic amino acids, and they can form pyruvate or bypass acetyl-CoA to enter the citric acid cycle. Part or all of that carbon skeleton that's left over after taking off the amine group can be used to form oxaloacetate, and we can actually use oxaloacetate to make glucose. There are other amino acids that are considered ketogenic amino acids because we can't use them to make glucose, but ketogenic amino acids can be used to make acetyl-CoA. They also can be used to make ketone bodies. So in gluconeogenesis, we're making glucose from amino acids. We can also make glucose from the glycerol part of fatty acids, but this is very, very inefficient. Gluconeogenesis occurs in the liver and the kidneys, and liver is the primary gluconeogenic organ. It starts with oxaloacetate, which remember is a 4-carbon compound, and it requires multiple vitamins as well as ATP, so biotin, riboflavin, niacin, and B6. Okay, so after we have deaminated an amino acid, what happens? Deamination removes that amine group, which is that nitrogen group. And that nitrogen group all right, sorry if this is a repeat, but my recording got interrupted. Anyways, we have to get rid of the nitrogen groups from amino acids, and so we do this with the liver and the kidney. We take off the amino group, the NH2, and we can convert it to ammonia. One ammonia and one amino group will react with CO2 to form urea and water, and urea is a much less toxic compound than ammonia. Then we will urinate out the urea in our urine so we can get rid of it. All right, what is this? So this is a picture of glucogenic and ketogenic amino acids. Remember, glucogenic amino acids can be converted to glucose, and so those are shown in green. Ketogenic amino acids cannot be converted to glucose, and they turn to acetyl-CoA. You don't have to know which ones are which for this class, but it is important to know that there is a difference. Okay, so this is a summary picture of gluconeogenesis, and amino acids that can be converted to glucose or can produce glucose are first converted to pyruvates. They directly enter the citric acid cycle, or they are directly converted to oxaloacetate. The ketogenic amino acids, those that can't yield glucose, are converted to acetyl-CoA. And then the glycerol part of triglycerides, remember our glycerol backbone, can be converted to glucose. All right, so here we go. So what is happening at the top of this? At the very top of this, we have glucose. And normally glucose is converted to pyruvate, moves to acetyl-CoA, goes through the citric acid cycle. But in the case of gluconeogenesis, the arrows are going the other way because we want to convert other things to glucose when we don't have glucose. So one of them is glycerol from fatty acids. Other things we can convert to glucose are glucogenic fatty acids. Fatty acids from beta oxidation can be converted to acetyl-CoA, but not glucose. And ketogenic acids can be converted to acetyl-CoA, but not glucose. So these two cannot be converted to glucose. However, they can still enter the citric acid cycle. Additional glucogenic amino acids will enter here below citrate, and more glucogenic amino acids will be converted to oxaloacetate, and then the arrows all lead upwards to glucose. Right, alcohol metabolism. So there is an entire chapter on alcohol metabolism that we actually skipped for this class, but you will learn about it in further classes in nutrition. So alcohol dehydrogenase is the main pathway by which alcohol is metabolized. And this occurs mostly in the liver, but a little bit in the stomach, maybe 10 to 30% in the stomach. And here you can see ethanol or alcohol being converted to acetaldehyde and acetyl-CoA, which can then enter the citric acid cycle. The thing with this is, in order to convert ethanol to acetaldehyde, you have to use up NADPH and you have to use up NAD+. So if you run out of NAD+, you can't do this pathway. Right here, this arm of the pathway is the primary pathway for alcohol metabolism using the enzyme alcohol dehydrogenase. In the case that NAD levels are not adequate, then instead we can use this second alcohol pathway, and this usually happens during high alcohol intake. So this is the microsomal ethanol oxidizing system. And this actually uses energy instead of yielding energy. In the first pathway, you're making NADH, but in this second pathway, sorry, my arrows are getting crazy here, you are using NADPH. This accounts for about 10% of alcohol metabolism. Catalase is a third pathway for breaking down alcohol and is considered a minor pathway. If NAD plus is limited in that first step, then NADH levels will build up and the citric acid cycle will slow down. And instead, we will use alcohol to generate fat and triglycerides. All right, so if consumption is moderate or low, so low consumption can allow small amounts of acetyl-CoA to form through the alcohol dehydrogenase pathway, and they can enter the citric acid cycle and produce energy. However, the breakdown of alcohol using this alcohol dehydrogenase pathway needs NAD, which I've just said, and NAD can become limited and then NADH levels build up, the citric acid cycle slows and blocks the entry of acetyl-CoA. Because the toxic effects of alcohol and breaking it down take priority over the continuation of the citric acid cycle, most of this acetyl-CoA that starts to build up is directed to fatty acid and triglyceride synthesis. And this results in accumulation of fat in the liver, which is called steatosis. This can be detected when a person has high circulating levels of triglycerides in the blood. If someone is consuming a lot of alcohol, we have that second pathway, which I showed you on the previous diagram, the MEOS pathway, or the microsomal ethanol oxidizing system. And this uses oxygen and a different niacin-containing enzyme and produces water and acetaldehyde. So this second pathway uses energy. The third pathway is the catalase pathway. Why do we care about fatty liver? Healthy liver can be transformed to a fatty liver in these conditions, and fatty liver is more likely to become a scarred liver or a cirrhotic liver when a person has cirrhosis. And a scarred liver is literally covered with scar tissue and does not work anymore. All right, regulation of metabolism. So the liver is one of the very important organs in metabolism, and it will respond to hormone and vitamins. So other regulation pathways involve ATP concentrations. Obviously, metabolism is going to slow down when we have plenty of energy. Enzymes, we need necessary enzymes to complete all the steps. Hormones, vitamins, and minerals. Remember that vitamins are involved in many of these steps. This is a summary slide of metabolism, of everything we have talked about today. And here you can see where glucose enters and can be used in the pathway. Above glucose is glycogen. You have triglycerides. Triglycerides break down to fatty acids and glycerol, so you can see where each of those is able to go. And then over here, we've got our proteins, which break down to amino acids and enter the citric acid cycle, become amino acetyl-CoA, or can become pyruvate over here. So Yes, this is a good summary slide for you to kind of remember what is happening. All right, what is the key organ to metabolism? The liver. So this is just another summary slide where you can see how the liver helps us break down and metabolize all of the components of foods that we eat. The liver is important for fat synthesis. It can convert simple forms of sugars. It can produce ketone bodies. It's responsible for amino acid metabolism, urea production, alcohol metabolism, and nutrient storage. So we're talking about regulation here. So we said many things regulate our metabolic rate, and one of them is ATP concentrations. So what would you think would happen if there is a high amount of ATP? Well, hopefully you would say we would decrease energy yielding reactions such as glycolysis and promote anabolic reactions such as protein synthesis. Remember that anabolic reactions actually use ATP. If we have a high level of ADP, that means we don't have a lot of energy in our body, so we would increase our energy yielding pathways. Enzymes also play an important role as key regulators of metabolic reactions, and the presence and rate of activity are critical. So presence meaning you need them. If you ran out of enzymes, whatever step that enzyme is needed stops. Hormones are important. Insulin, for example, if you have low levels of insulin, say you're fasting or not eating carbohydrates or maybe even diabetic, this is going to cause gluconeogenesis to be activated. So you're going to break down proteins and break down lipids, and then that will increase your glucose levels. If you have high levels of insulin, perhaps after eating carbs, you are going to make glycogen to store those carbs, fat, and protein. These are the vitamins and minerals that play a role in metabolism. And you can see why, if you have any nutrient deficiencies, parts of this metabolic cycle, metabolic pathway, could slow down or even stop or not be possible. So yeah, for example, here we've got vitamin B6, and it is the only vitamin at this step. So what if you had no B6? Would you be able to convert carbs, glycogen in particular, to monosaccharides? No. Here we need niacin for alcohol conversion. You see niacin a lot. Remember, niacin is part of NAD, NADP. We've got iron, we've got folate, we've got riboflavin, copper, biotin, B12, just so many vitamins playing a very important role. Okay, what about fasting and feasting? What happens to nutrient metabolism? So these are diagrams from your book, and I don't love them. They're okay. Yeah, they're okay. I don't love them. What they're supposed to represent, what they're supposed to represent is what happens in fasting. And the way they're supposed to represent that is by the thickness of these arrows. So if the arrows are thick, that means that that pathway is being used a lot. If the arrows are thin, that means that that pathway is not being used that much. In this very top diagram, I don't think that this protein arrow should be quite this thick. That's what I don't like about this. I think it's a little too thick. Okay, so in the beginning of the fast, you are going to use glycogen. Remember, we have glycogen stores in our liver and muscle, and we can break those down. We can also use fatty acids from adipose tissue. Eventually, the liver glycogen stores will be exhausted, and to provide much needed glucose, we can actually start breaking down our protein and using those glucogenic amino acids to make more glucose. So what are some of the longest fasts? These are just fun facts, but I thought this was really interesting. So during a prolonged fast, the body protein will continue to be broken down. Can we use fatty acids for gluconeogenesis? No, we cannot. We can use glycerol, but not fatty acids. Sodium and potassium will become depleted. They are drawn into the urine with ketone bodies. Ketone bodies will build up. Remember, if there's no carbs to make oxaloacetate and to enter the citric acid cycle. So if there's no oxaloacetate, then acetyl-CoA from fatty acid breakdown has nothing to combine with. Ketones build up, cause increased urination, draws out electrolytes with it. Eventually, death can occur, and death can occur no matter how much body fat a person has, because we can't use fatty acids for gluconeogenesis. So eventually we will be breaking down our protein, and once you've broken down about 50% of your protein, you die. What else happens? So blood urea levels increase because we're breaking down protein. This is a picture of the longest fast ever. Sorry, I take that back. This is a picture about somebody who did fast, and he was doing a fast as a protest. And I just think it's really interesting, that he fasted so long. He fasted for 73 days. However, there's somebody who has fasted even longer than him. In 1973, there was a 27-year-old male who underwent a medically supervised fast, and he fasted for 382 days. No food. That is insane. How did he do this? So it was medically supervised, like I said. This man started at 456 pounds. When he was done with the fast, he was 180 pounds. So he lost 276 pounds. He was using those pounds as his energy reserves. He was given water. He was also given vitamin supplements. And during the middle of the fast, he was given sodium and electrolytes, because if sodium and electrolytes were totally depleted, sodium and potassium, he would die. So he was given those things and monitored carefully. Interesting, he had no bowel movements or very, very infrequent bowel movements. His bowel movements occurred on average every 37 to 48 days. So can you imagine going a month without a bowel movement? Just think that's crazy. How he could survive so long. So we're going to get to this. So our body does try to help us survive. It's an adaptation, and we will slow down our metabolic rate and reduce the amount of energy we need. This helps us slow down the breakdown of our protein, our lean tissue, our muscles, to supply glucose for gluconeogenesis. Another adaptation we have allows our central nervous system, our brain, to use less glucose for energy and rely more on ketone bodies. So in the beginning of the fast, the brain isn't really fond of ketone bodies, but it can adapt to use more ketone bodies. And we can produce ketone bodies by fat breakdown, so we can start using more of our fat. However, like I said previously, once we get to a point where we've broken down so much of our muscle, we will die, unfortunately. Feasting. I think you guys are probably more familiar with feasting than fasting. And the most obvious result of feasting is fat storage. Feasting encourages insulin production by our pancreas, and that encourages the burning of glucose for energy, as well as synthesis of glycogen and protein. Okay, where does fat go that is consumed in excess? Where do you think it goes? It goes to the adipose cells. So fat will go directly to the adipose cells. There is little energy needed to store it. It's a very efficient way to store fat. Eating fat is easy to store as fat. carbs that are consumed in excess. Carbs that are consumed in excess can be used for glycogen, or they can be used to make fat. But it's not as efficient as a cycle or a pathway as fat to fat. So converting carbs to fat is a little bit harder. However, we can still do it. Where do proteins go that are consumed in excess? So first of all, they go to amino acid pools. Remember, we said we can draw from our amino acid pools. However, the amount that goes to the amino acid pools is not that much. It's pretty low. Next, we can use proteins to make fatty acids, but again, we don't do this very often. It's not usually necessary. And then third, we can use proteins to make fat, but this requires ATP and B vitamins, so there is a very high energy cost in converting proteins to fat. So you should know kind of which macronutrients store as fat easily versus not. Track the nutrients. All right, you've seen this already, but this is when we eat nutrients in excess. Where do they go, or where can they go? Metabolism of ATP yielding compounds. I think this is a great chart to help you remember where everything goes and what it can be used for. So you can see that fatty acids are the easiest to convert to fats. And that's it. That is the last slide. So thank you for listening with me today. I know metabolism is a little bit in depth of a subject, but I hope I helped in your understanding, and I'll see you next time.